Amniotic fluid may hold the key to growing human organs in a dish. Let's talk about it. Stem cells are a special kind of cell that are capable of making copies of themselves that turn into different cell types, or differentiate. Most of your cells are at least semi-differentiated, meaning that they're at least on their way to becoming a specific kind of cell with a specific job, and they can't change career paths. For example, the stem cells in your bone marrow are capable of replicating into red and white blood cells, but those blood cells themselves cannot make any more copies of themselves nor turn into any other kind of cell. They are terminally differentiated. Stem cells are incredibly useful in medicine because they represent a source of other cell types that a sick person may need replaced. As you might imagine, it takes a lot of stem cells to build a human being completely from scratch, so embryos are chock full of them. And because they hang out in amniotic fluid all day, sometimes some of those stem cells will break off and start floating around in that fluid. If you extract a syringe full of amniotic fluid, chances are there will be some stem cells floating around in it, which means that you can get embryonic stem cells for research without having to touch the embryo at all. You just bottle up some of their bathwater. Now, this in itself isn't new, but last year a group of scientists reported that they had figured out a new way to turn those stem cells into structures called organoids. Organoids are basically miniature 3D models of organs, and this research showed that scientists could take as little as one single stem cell from amniotic fluid and grow small intestines, kidneys, and lung organoids in a dish. Now, can you grow a whole functioning baby organ from a single stem cell taken from amniotic fluid? No. Or at least not yet. But this was a really important step towards that, and it's also super useful on its own. See, organoids are crucial models for figuring out the underlying mechanisms of how diseases work and progress, which is the first step to coming up with treatments. And the organoid's predictive power can inform how current treatments should be used. Right now, embryo testing is really good for telling you if a baby will have a disease, but not necessarily how severe it will be or what kinds of specific medical care doctors need to have on hand the moment the baby is born. But an organoid in a dish is a predictive model. It can show you how bad a disease will be and how quickly it will progress based on how it behaves in the lab. And these predictions provide essential clues to doctors and parents about how to best prepare for a baby who might need a little bit of extra care.